Thank you, everybody, for coming to the seminar. We have Graham here, who's going to present on his really cool work, as is indicated by the title. He was educated at the University of Leeds. Do you want me to give the date? In the 80s.
They can figure out it was a while ago.
In biochemistry, a BS in biochemistry, followed by a PhD in biochemistry from the same place. He was actually here at Dartmouth from 1999 to about 2002, where he was a research assistant professor. And then he was a senior researcher at the University of Wales College of Medicine before moving to a completely different climate and becoming a full professor at the University of New Mexico in 2000. Started in 2001 and then ultimately went through the rest to become full professor. So Graham is also an entrepreneur in addition to a researcher. So his research area you'll learn more about. It involves doing some really cool things with stable isotopes to diagnose disease and track the efficacy of treatment. In addition to that, he's an entrepreneur, as I was mentioning. He's a co-founder of Avisa Pharma, which develops stable isotope breath tests for lung infections. And he's also the co-founder recently and chief scientific officer for Specificity, correct, which is developing labeled TB drugs for TB, drug-resistant TB. And now he's doing that now full-time, taking a little bit of break from the academic world. So with that, take it away.
Thank you, Jane. And I never made it to full professor, mainly because all this entrepreneur stuff takes you away. Disclosure statement, obviously, for lots of this stuff, I got skin in the game. I was saying to some of the students, I managed to buy a used Mercedes already off of some of the money I've made. Make sure you realize that I stand to make some money from this, so it's not just academics as well, although everything should be good. What I want to talk about are this breath test stuff, and really 4 different parts. Breath test detection, why we might want to do it, how do we do it, give you some examples of the different chemistries, then talk about the problems when you try and take this from in vivo through to in the clinic, some of the unique problems, some future projects and some general concepts, some of the things we've learned, as well as early success, which kept us going through this. We've also had some quite epic failures as well, and so what have we learned from the success and failures that might guide how Jane and collaborators might want to use this kind of work. So the first one is the overall concept of bacterial phenotype detection. Okay, so if you think about old-fashioned microbiology, this is what you might see. These are a bunch of different compounds. The bacteria is grown, ferments them or transforms them in different ways to give a pH change or whatever. And so from the different patterns of these colors, you can look at, okay, this bug is capable of fermenting glucose or indole, it has Yura Gaze or wherever. And so you've got to get a sample, you've got to grow it, but it detects and differentiates the species by metabolic profiling. And this, before sequencing was so easy, was how clinical microbiology was done. Now, it's all genotype-based, okay? So you get a sample of nucleic acid, and sequence is very, very powerful in telling you what species are there, okay? We're more interested in point-of-care detection. So this is actually one of our competitors. You can see it's a rather large unit. So the idea of putting this on the back of a bicycle and going around villages in Africa to diagnose TB is probably not going to work. There are much smaller versions, obviously, but I always show this because it makes us look good. Well, what I want to do is, okay, we've got this really old-fashioned microbiology that's been going on since the 19th century. How am I going to get you into this? How am I going to get you to believe that we can do this high-tech metabolic testing, okay? And what use is it going to be, even if we can do it? And this is the overall concept. And does everybody still remember clip art that you used to get in Microsoft Word? Because it's gone away now, I've noticed. It's not there anymore. But I always used to say to students, if you can't explain your thesis in one slide using clip art, it's too complex. You've got to simplify it. And this is essentially it. So this is, obviously we're in Albuquerque, so this could be a meth pipe. In this case, it's actually an inhaler. So we have a patient who's going to inhale a substrate. So here we have this substrate, and it's going to be stably isotopically labeled. No radioactivity. We don't need to worry about radioactivity. It's going to go essentially everywhere where the lung is still functioning. So we're going to sample most of the lung, if not all of the lung. And if we've got a bug or an infection here shown by this little green ball, it's going to convert this into a labeled product. And again, we're interested in gases. If it was if it didn't have a high enough vapor pressure, we wouldn't really be interested in it. And exactly this, it's very analogous to what Jane's group does. So we're going to have a chemical transformation. It carries its label with it, and then it's exhaled out. And then we can either look just for this, or in some cases we can look at the ratio of labeled product against normal product. So a lot of the work we do is in CO2. So even though we exhale 5% CO2 at the end of our breaths, we can still tell the difference as to where it came from because of the isotope ratios. And the idea is, from these isotope ratios, we can understand the presence of bacteria in the lung. We can understand the site. If the bacteria are in the airway, the signal comes out very early in the breath. If they're deep in the alveoli, it comes out very, very much later. So we can look at the site, and clinicians tell me that this is important. We can look at load. We can look at response to treatment. And we can also look directly at resistance as well. So we can, if this treatment is working, we'd expect the signal to go down rather rapidly. But there are some ways we can directly look at resistance. So why do we want to do this? The sequence is becoming established and very, very good. Sometimes it can be difficult. What you need, though, is a sample from the lung, and sometimes it can be difficult to impossible to get sputum. There are different procedures to get induced sputum. Sometimes people just don't make sputum. So if you're interested in cystic fibrosis early on, younger patients don't make sputum, so you can't get this analyte. And even if you do, what does that represent? Does it represent the whole lung? Does it represent one little corner? Is it the worst part of the lung? You don't really understand the entire lung. And there are more invasive things you can do. You can do bronchoalveolar lavage, where you stick a tube down into somebody's lungs, spray liquid in, and pull it back out again. And it's as invasive as it sounds. I wouldn't like to have one of these done. Induced sputum, where you nebulize in, usually hypertonic sodium chloride. So you just make people cough really, really hard. It's not so bad. But if, let's say, you're in, you think somebody's got TB, and you've just induced them to cough deeply and hard for the next 10 minutes, I don't want to be in that room while that goes on. I've seen it done in South Africa, and they do it outdoors. So they have the separate induced sputum hut, and it's about 50 feet away from everywhere, open to the elements. Somebody goes out there on their own and does this and produces a sputum, then gives it back, and everybody stays away while this process goes on, okay? So it's not always safe. Why would we want to do breath? Well, we get coverage of the entire lung. So here you think it goes in. We're not filling up the lungs with liquid here, okay? That would be really bad. But what we're showing is that we can look at most of the lung, and it's relatively easy to do. And as well, sequence is not the be-all and end-all. So sequence is really only useful when it absolutely indicates disease. And for lots of pathogens, we don't know, is it like Staph aureus? It's part of our transient flora. Lots of people are colonized with Staph. So if you measure somebody has a pneumonia and you measure a Staph DNA, then it may not necessarily tell you as much as you might think it would do, or you might hope it would do. For some things, like sequence for TB, let's say, sequence really is indicative of disease. So for lots of disease, it's great, okay? I'm not trying to knock sequence. It's just that for lots of the difficult to diagnose lung infections, it's really difficult to do. And if you think about it, the lung has an area of, well, probably about this. The tennis court is what they say. And it's in intimate contact with the external environment. So we breathe in all this air. And you would think it would be easy to diagnose stuff and yet, from breath, and yet people are really only taking this seriously recently. For other things as well, so for some drug resistance, a certain sequence, so you detect a mutation in an antibiotic target, sometimes it's absolutely, you can say, yeah, that's going to be resistant. Often in other cases, it's much more ambiguous. And like I was talking to Joe, like pyrazinamide is probably the worst case where you get sequence of from TB. It's really not clear whether that's going to be drug resistance or sensitive, and yet the phenotypic tests, difficult though they are, can usually pick this up. So often there's not a strong linkage between drug resistance phenotype, which is what we're really interested in, and the genetic sequence. And assuming you can actually get sequence as well, sometimes the sampling is just too difficult. So what are we going to do? How are we going to make this isotope breath test kind of work? Well, the first step is we're going to need specific biochemistry and detection. So can you make the tracer? Ideally, can you buy it? Because although I make stuff, it's more work. Can we detect the product? Okay. We need, because we're relying upon bacterial enzymes in situ within the patient, is there enough processing of this tracer to actually be able to detect it? So if you are making labeled CO2, you're going to have to compete with a large amount of exhaled CO2, all of which contains 1% carbon-13 CO2 as well. It's got to be fast as well, because the lung is, as we saw with meth pipe Mary, the lung is a great place for absorbing compounds, okay? That's why people, when they're abusing drugs, their ideal choice is either inhaled or intravenous, because it hits the central nervous system very, very rapidly. So the problem with that is we're going to get systemic absorption over a period of maybe 20 minutes. That's going to hit the gut microbiome, and all kinds of stuff is going on in the gut. It's a very, very complex microbial environment. And for lots of the tracers we're interested in, especially urease, the gut is just full of urease. So you've got to be able to have this rapidly enough so that you squirt the stuff in, and then 5 minutes later you've got a signal. Because if you leave it half an hour, then you're going to be looking instead at what the microbiome is. So you really want to have fast chemistry. And the tracer needs to be inhaled. So this leads to questions of toxicity. I won't go into reasons. FDA never really looked too closely at inhaled compounds. Now they do. So you've got to think about GMP material. You're going to have to do GLP tox studies. So as you'll see, we get a lot of specificity. We can have very, very high specificity because we're just looking at the conversion of one labeled compound to another. A big cost of this is the money and the regulatory aspect. And also, we also want to avoid cough, which is generally easy to do. If the compound isn't noxious, then if you give it as an isotonic nebulized solution, usually it's well tolerated. That's what we found. And this is small enough. If anybody has any questions, just put your hand up. I don't mind stopping. Okay? So how does it work? Well, you get people to look on the internet along metabolic pathways and search for the volatiles. So you look at a metabolic pathway and you tell a student, okay, search for CO2. And obviously that gives you tons of stuff. loads and loads of decarboxylations occur biologically. And then you tell them, okay, search for that metabolism in every other potential pathogen that we could have. And, we're interested in lung infections, so all escape pathogens, anything that could be is known to be a lung pathogen. How specific or non-specific are we? Okay? And you figure out what's your sweet spot of specificity or non-specificity, as you actually see for some of our stuff. Then you've got to think, well, can we make this? Is it possible? Is it feasible to make it in? Can we buy it? First of all, you can buy it. It's good. If you have problems that money can solve and you have the money, you don't have a problem. But we're going to have to make it. And then how are we going to detect this? Okay, is it going to be possible to actually detect this? And we tend to just use isotope ratio mass spec, which is can detect a very, very wide range of isotopically labeled volatiles. Okay, it's a great big machine, so it's not portable. We're not in point-of-care detection with this. But the first scan we want to see, is it worth going any further at all? So this is actually usually relatively easy. Then we'll do some in vitro testing, exactly the same as Dr. Hill's group is. You'll get a range of pathogens, grow them in tubes, put the stuff in, and then look for the labeled compound or the labeled product in the headspace. So usually, because it's a very, very concentrated broth, and you'll give them a period of time, you should expect huge signals. If you're only seeing marginal signals at this stage in a highly enriched bacterial culture, don't proceed any further. Okay, you're just making trouble for yourself. And so you can look at specificity. You can look at all the lung pathogens you're interested in. Is this specific or not? That doesn't tell you it's going to work in patients, because those isolates may behave quite differently when they're in the lung environment than when they're actually growing on some kind of broth or whatever. The processivity I talked about, is there enough of a signal to actually be worth working with? Then you need some in vivo testing. So you need a good model. And lots of our work in TB has been done with Bill Bishai's group in Hopkins. Rabbits are just about big enough so that you can trach them and collect gases and put stuff in very, very easily. It's difficult to do with a mouse. We also get big gas samples, and yet they're not so big that we have to make loads and loads of tracers, so we can get away with relatively rare compounds, and that's worked very, very well. And then if everything looks good, then you can think about going clinical. You're going to have to raise some money, because this stuff is expensive once you go clinically. And I don't know if anybody here has figured out a way of getting clinical trials funded through NIH so you can get all the GLP and GMP stuff done. It's not very innovative. I've never had success. I want to give you some examples of kind of things we've done. Balancing sensitivity and specificity, and what have we learned, if anything? I always like that movie. Do you remember Burn Before Reading? What did we learn? Don't let it happen again, okay? And sometimes that's all you're going to learn. The first thing we were interested in is Yura Gaze, and this is a classical discriminator in clinical microbiology. So people will Yura Gaze positive, Yura Gaze negative. And it's just part of a broader metabolic discrimination between species. But we thought, well, what if we can actually do this in vivo? So the first thing we did, take urea, and it cleaves CO2. It cleaves it to carbon dioxide and ammonia. We've tended to look at the CO2 because there's a FDA available detection technologies. However, were I to do this all over again, I would probably look for the labeled urea, okay? Ammonia is a much rarer gas in breath, so the background is much smaller. So, that's what I would have done. It's a common bacterial virulence factor. It's already present in the airway surface liquid. So we already know it's there about 2 to 5 millimolar. So when we speak to FDA about likely toxicity, we already know it's always there in the lung at a relatively high concentration. So they can have a good feeling about the toxicity of this. You know, it's certainly at those levels not chronically toxic. And it's already used clinically for H. pylori detection. So Helicobacter pylori won Barry Marshall the Nobel Prize. I think it was 98. And he was the crazy Australian who showed that this bug causes ulcers by growing it up and drinking it. And then getting endoscopy and shown he actually got an ulcer and then sampled that ulcer. Now, it's not exactly IRB compliant, I don't think. But things were kind of different then. And good for him. I think this kind of gonzo science is to be applauded. And so they started with a breath test. They were using radio-labeled urea. And so carbon-14, a small amount. So the idea of this breath test is very old. We just use carbon-13. But because this is already used for pylori, we call it an API, an active pharmaceutical ingredient. Although it's not really a pharmaceutical, it's not clear what we should call it. So we call it API. It's available at GMP, which means it's a high-quality product that is safe to go into people and has met a number of thresholds. And this, making stuff at GMP, is expensive. There's an FDA-approved detection device. So it's a classic dosage route repurpose kind of thing. And down in UNM, we're really big on repurposing drugs, using drugs that are known to treat other diseases. So everybody thought, this is great, nice and easy. Our first in vivo experiment, obviously everything worked fine in vitro. We proceeded. To give you an idea what goes on, this is a poor rabbit being infected with TB. And you can see that the person doing it takes a lot of care not to get TB themselves. Here's the rabbit. It's got some anesthetic. And so we put the 13C in and collect the breath. And probably what we were most interested in, because as I'll talk about, urease is very non-specific. But at the time, we were trying to look for a treatment biomarker. So can you use the up and down of the urease signal to tell you whether your antibacterial agent is working or whether it isn't? So here's data from one rabbit. Here's pre-infection, and in white is the 13C signal. And in gray is CFUs from bronchoalveolar lavage. So at each time point, the rabbit had bronchoalveolar lavage, and CFUs were plated out. And we've normalized these to 1 post-infection. Okay? So here you can see no CFUs. Worryingly, we have a little bit of a background signal. Post-infection, whopping great signals on both. Then we treat the rabbits with isoniazid for 2 weeks. This isn't clinical therapy. You would never use monotherapy. You would never use isoniazid on its own. But you can see we get nice decrease in the bronchoalveolar lavage, and again, a nice decrease in the urea signal, okay? Then when we withdraw isoniazid, so take away the isoniazid treatment, so let's say simulating the development of resistance during therapy, the CFUs bounce back, the urease bounces back. So we can see when we kill off the bug, the signals go down. When we let it grow back, we get these signals. Now, there isn't a perfect match between these, obviously, but at least the trends go the same. So you could use this to inform treatment decisions. The problem is almost all non-viral lung pathogens make urease. So there's really only strep pneumo of the major ones, and a few of the Enterobacteriaceae that don't make urease. But you can see all the ones we're interested in. Escape pathogens. Here's, everybody's heard of escape, ESKAPE. So here's Acinetobacter, yeah, Klebsiella, Pseudomonas, Staph, Cryptococcus neoformans. Haemophilus influenzae isn't one of the escape pathogens, but is a significant cause of pneumonia. And you could look at lots and lots of other bacteria. So valley fever and fungi, valley fever, aspergillus, they all have Yura Gaze. There are plenty of other bacteria as well. So it might be a good non-specific thermometer. But if you're going to use this to try and achieve speciation, so you know there's something wrong in the lung, and there's one of these pathogens that's there, but that doesn't enable you to design a therapy. You don't know what you're going to use to treat it, so you're really almost still in empirical antimicrobial therapy. So we haven't done much. But it is a good thermometer. So you might be able to longitudinally monitor treatment and see if it's appropriate or not. So we wanted to go to more specific chemistries. And one of the more interesting ones was this CODH, carbon monoxide dehydrogenase, okay? And it's expressed by TB. And you think, well, what is this doing? And this is an enzyme that will take carbon monoxide and oxidize it, okay? And if you go into the literature, you can see that you can actually grow TB on carbon monoxide, the sole carbon source. It's phenomenal. And when you think about it, it's just a relatively recent soil bacterium where there's lots of CO in, let's say, volcanic seeps or stuff like that. So it's had the opportunity to develop this unique chemistry that then, once on becoming a pathogen, it brought it with it. And it must, it's probably there for a reason. We know that there's a big CO impacts TB a lot. So if you, it's probably doing something. But it's very rare amongst lung pathogens. We couldn't find this either annotated or unannotated in any other lung pathogen. So we've got the possibility for very, very high selectivity. And what you can see is we've got a double label here, okay? So we're going to use double labeled 13C, 18O, and we're going to have the double labeled CO2, which is labeled as mass 37. So if you look, so you can see what some of this in vitro data looks like. Here's a dose, how much CO we put in the tubes. Here's how much of the signal we got in the headspace. Here is a CFU sensitivity. So we add increasing amounts per milliliter of the bacteria. And you can see you get an increase. It seems as if it saturates here, okay? Which isn't really what we want to see, and we don't understand what's going on. But that's what we've got. But there is at least a range over which the signal appears to be proportional to the amount of bacteria.
Is there a number of amount of time that concentration is associated with?
Yeah, so the longer you leave it, the more you can detect. But you can't, we only think about, if you can't get it done in 10 to 15 minutes, the gut microbiome comes into play. So if we can't get a big stonking signal in 10 to 15 minutes, we don't think it's worth moving any further. And this is the data from rabbits. So this was a heroic study. This was exceptionally difficult to do. And you can see here are controls in red, time after inhalation, and here in black are the infected, and you can see there's a time-dependent increase. So we could differentiate between these two. And you might be thinking, well, why are you can't give CO to people. You can't use that. Actually, you do. So there is a pulmonary test called diffusion capacity that they will give or an inhaled bolus of carbon monoxide and then look for the appearance of that. And from that, you can look at the lung diffusion capability. What really killed us is this double-labeled stuff is really costly. And so when we scaled it to what it might be in humans, especially with TB being kind of like we don't want expensive stuff for TB. Just put it that way, the market for expensive $100,000 TB diagnostics doesn't occur. So we've actually thought about using accelerator mass spec. So if we gave 14 CO, could we do that? We just haven't found anybody interested who's capable of doing accelerator mass spec. But for sure, you would get absolutely excellent discrimination, and it's very, very specific. Well, we breathe out CO all the time. CO is made by hemoxygenase, amongst other things. We were only looking at the ratio, so, but we never directly, I'm trying to think. Because we're only looking at the double-labeled compound, that any contribution from host CO being metabolized to CO2, we just don't see that because it's the amount of double-labeled, naturally abundant CO is fleetingly small. So we just can't see it. We only see what's coming from our tracer. Did that answer the question?
Sorry if this is coming up, but have you assessed your sort of ability to detect burn in vivo?
Not with this. We've done it with Yura Gaze, and that's all we've done it for. No, for the next one, we also look at this. Another one where we're looking at CO2 is tularemia. And tularemia is caused by Francisella tularensis, and this has a rare virulence factor called citrulline ureadase, CTU. And this takes citrulline and fragments it down, giving you potentially labeled CO2. And why this is a virulence factor is this is the product of nitric oxide synthase. And then what usually happens is citrulline goes back, gets converted back to arginine. And so you keep on making nitric oxide. And this nitric oxide is really important in controlling Francisella tularensis in the macrophage. But this gets rid of that recycling. So the nitric oxide levels go way, way down, and it's able to survive nicely in the macrophage. Now, the problem with this is a select agent, which means if you're going to do this work, the FBI are going to want to know why, and they will come and inspect your lab and your people and all that kind of stuff. And that meant that our usual sterilization strategies, which just involve double membrane filtration, that wasn't good enough. We had to validate that the samples coming out of the select agent lab truly are absolutely free of live Francisella tularensis. And conceptually, a gas sample is very, very difficult to sterilize. We did, through an autoclave, we did try it, and we could never find bags that could withstand 15 psi, 121 degrees centigrade. These are all polymer bags, and everything either exploded or collapsed or melted or combinations of that. So we ended up absorbing it onto Sodasorb, which is the same stuff that you use to absorb CO2 in anesthesia machines or rebreathers. And so we can absorb this, chemically absorb it, autoclave it, and it's still stable. But then when we put this, we take the Sodasorb out, we put it in the elemental analyzer of an IR mass spec, and we drive off all of the CO2 again. So we can do that. It's a nice trick. So this is the mouse, and this is some relatively recent data. So here is they received the citrulline. Here's the baseline. You can see for this mouse, nice big peak in the first sample, which actually represents 0 to 30 minutes coming down. And you can see that for 3 different mice. So it looks like we might be able to get somewhere with this. And we were really hoping, with all the biodefense money that's around, that people would get really excited. However, when you're dealing with people like DARPA, we say, oh yeah, we can measure this one thing. And in their ADHD-like manner, they want the tricorder. They really do believe that Bones is going to be there with the tricorder and be able to scan and say everything all at once at the same time. So we haven't got them to give us any money yet. And this is expensive work. So until we get more money, we're not going to do this. So what do you think is the problem with measuring 13 CO2 ratios in breath? Yeah, background. So the problem is, of all the breath CO2 that you're breathing out, 1% of it is already 13 CO2 anyway. And that ratio constantly changes. Because if you, let's say, just ate a corn tortilla, which is what we will eat a lot down in New Mexico, corn is a C4 plant and is very enriched in carbon-13. So if you just eat that and start metabolizing it, your 13-CO2 levels go up. And there's actually a breath test for gastroparesis, or stomach emptying, that's entirely based upon giving people carbon-13 labeled food and looking at how fast those levels go up. So you have this constant varying background of 13-CO2 at all times. And the ratios can change by an awful lot and swamp your signal. So it's although most of our work has been in this, looking back, it was kind of a bad analyte. We should have been looking at rarer things. So let's try and study some of the rarer gases. We've had a big, long interest in isoniazid for years and years and years. And it's a prodrug. So this is what isoniazid looks like. Very, very small compound, but exceptionally potent against TB. That sounds like, do you remember Forbidden Planet? Does anybody remember the sound from that? I forget what it was, but it's very reminiscent of Forbidden Planet. And it's metabolized by a specific TB enzyme called CatG. It makes this radical and also makes this compound, diazine, okay? Not ordinarily what we would expect to see around. And this reacts with any double bonds nearby, reducing the carbon-carbon double bonds and making nitrogen. So if we label both of these with 15 nitrogen, then we get labeled N2 gas out at the end. And double-labeled N2 gas is exceptionally rare. So although you're breathing out, 70%, 78% nitrogen all the time, the fraction that's actually this double-labeled variety is tiny. So although you think of nitrogen as being very abundant, when we're looking at this, it's actually very, very rare in breath indeed. And so it enabled us to really drive this up. And again, this is quite specific. Here's the CFU dependence, here's dose dependence, and here's incubation time. Here's some specificity. So here's the amount of signal that we get for TB, and here's some other lung pathogens, and these are all, not statistically different from zero, so we can tell the difference. And also, what we're most interested in, the high-level resistance against this compound is due to mutations, especially this S315T mutation in the CatG, so it no longer activates the drug. And when we look at wild type against resistant, we can totally tell the difference. So we think we should be able to do if we could measure the nitrogen in a point-of-care manner, we could do drug susceptibility. And if you look at the rabbit data, this is rabbits infected with a high dose. This is rabbits infected with a low dose. Time after we've put this compound in, you can see after 5 minutes, there's this monstrous increase in double-labeled nitrogen. It could be in as little as a minute. We just never went that short. We never expected it to be so good. And 5 minutes is good enough anyway. In some rabbits, it comes back down. In the controls, you don't see anything. With a low dose, again, you can really discriminate between both groups super easily.
Have you established the specificity of the CatG enzyme? Like, we look at CatG mutants, we try to see other enzymes
Can compensate for the... No, it's well established. So we haven't done it, but we didn't get dinged in review. People still... It's not thought that there are other enzymes that can compensate and so metabolize this drug. So that won't be the case for everything. We're just in a lucky situation. If you look at... and it shows good dose dependence. So for some of these guys, we did actually get lung CFUs. For a couple of them, immediately after we've treated them. And you can see for the controls, there's nothing. For these guys, there's some kind of trend. I've drawn it as linear, but really when you got these 3 points, you can draw a lot of shapes. So whether or not we're justified or not, I don't know. If we look at the effective dose used, And the end CFUs should be a function of that given the infection time scales that we used. It was much better, so you can see that for a bunch, this was really, really quite tight. And so this shows CFU dependence. So for Yura Gaze and this is the only one where we were shown they're in vivo. The signal truly does indicate burden. And what's really interesting is the impact factor that we published in kept on going up, okay? So, and it's usually the case, your first paper is your best, and after that, there's this, dependent upon your stature within the field, there's a decay curve on impact factor, okay? If you're really famous, you can keep publishing in relatively high journals. If you remember the academy, you get 3 PNAS papers a year that you can contribute. For the rest of us, though, there's this relatively fast decline as it goes from being very innovative to, oh yeah, we've seen this before. But we kept on going up, okay? Maybe people didn't believe we could go anywhere with this. But that first one, which was a great paper, the best we could get was +1. That's how bad it was. I mean, these days, fortunately, we have a journal called Journal of Breath Research, and the impact factor of that is quite high. So I'm going to start using that a lot. Then it was MBio, then Nature Communications, and then Invited to talk about it in Trends in Micro. Usually it goes the other way. So maybe it was just the fact that people didn't believe us, or we weren't making enough of a noise. You know, one paper isn't really enough. It requires those kinds of things. And next we're going to talk about translating this to clinical trials, the regulatory hurdles, and some clinical data. So it should be really, really easy. Urea's natural product. We've got GMP, FDA-approved detection and delivery. Just nebulize it in. That was our naive, very naive hope. And it's wrong. There are actually very, very considerable regulatory hurdles in going from an oral to an inhaled dosage form, mainly because a subject, this happened when it shouldn't have done, and a healthy subject died at a major research university in the US. So FDA now looks at this very, very closely indeed. And so there's a lot of regulatory stuff. So this goes under a breath test, so it goes under an IDE, so the usual kind of regulatory approach that you might think of devices rather than drugs, but you end up producing huge stuff. The first trial was with Hengame Raisi. Darn it. Okay, I didn't realize that was going to come back on. I thought I turned off the projector. Okay. And we did this for Pseudomonas aeruginosa colonized cystic fibrosis patients. Why did we go with CF? Well, for the same reason Jane's group do a lot of work in CF. They're a stable population with well-characterized microbiology. And especially for us, because we're administering an agent to the lung, we didn't want to go in very sick people, because if you do very sick people, like in severe pneumonias, some of those people are going to die on your trial. And then figuring out, was it a problem related to our compound, or did they just die anyway? It's not a great place to start. So could be very, very difficult. And they got well-characterized bacterial stuff. And as well, there's a potential for it to actually be useful. So we thought, well, we might be able to look at microbial changes as a result of antibiotic therapy. Lots of people take stuff. Maybe there'll be a change that will predict an exacerbation. So we inhaled it in 20 mg and 50 mg total doses. And if you're wondering where does the dosing itself is a total art. The reason we went with 50 mgs is the cup on the nebulizer is 3 mils. And 3 mils to make it isotonic gives you just about 50 mgs of urea. So that was as much as we could go and be isotonic. And obviously we added some sodium chloride in a lower dose just to see if there was dose dependence. The breath was collected and it was well tolerated, no coughing, no nasty stuff going on. So here's some of the data from the 20 mg dose. So the healthies are in dark. We get a little bit of a signal. Here's time after nebulization. And And here with the CF, we can see some diversity, obviously. We wouldn't expect the signal to be all the same, but we could discriminate really, really nicely. Here's just the mean of that. Then when we look at the dose dependence, and here, I'm sorry for the relatively complicated way of looking at it, but it's the CF mean minus the healthy mean. It was the only way I could get all this data to really fit nicely on a slide. So you can see, nice, here's the 20 mg data you saw going back down, exponential. And here for the 50 mg dose, you can see just exactly the same kinetics, but more of a signal. Yeah. So they could be, certainly the ones we would worry about would be staph. So we can't say it's Pseudomonas specific. But if you're looking at, bacterial burden, you could do something with this. It's easier than doing, lots and lots of quantitative CFUs, for sure. And then can you see this stuff? We ended up in TB in Durban. And early on, so this is the receiver-operator curve. So the more that this thing goes above the diagonal, the better, for those of you that aren't used to doing it. And so this started looking really, really good when we just had 16 patients. And here you can see here's box and whisker plots between TB positive and TB minus. But the specificity disappeared as we did more and more patients. This was just a statistical fluke, really. We think we just got lucky with patients in the first group. And why is this? Well, when you look back, at the very least, you should learn from your mistakes. The control group in Durban had these big signals. So if you look here, these guys are making, peak signals of up to 25. And our UNM controls were kind of like 1 or less. So what was the problem? Well, the way that this was designed, we wanted to have a gold standard for TB. And the way you do that is to look at sputum, either by culture or by sequence. And we chose sequence because it's rapid. But our control group, therefore, had to make sputum. And sputum is not just spit coming out your mouth. Sputum is essentially pus that comes from the lung. So the same kind of stuff, you get a splinter in your finger, and then it gets all infected, and you can squeeze it out. That's what sputum is. So if you are making that in your lung, you are not healthy control, okay? And as we already saw, almost all of the lung pathogens that are likely to be there express urease. So we never had a problem with sensitivity. We could get everybody. It was excluding. It was the specificity that was the problem. But that's because we didn't have healthy controls. It's interesting to look back. The company has since moved on, is looking at something else. Where are we going to go in the next few years? Well, actually, we did this. So we were interested in pyrazinamide, another TB drug. And prodrugs are very, very common in TB therapy, as opposed to most other infectious diseases. So we were thinking, okay, we'll label this with nitrogen, we'll look at ammonia, and we'll be able to do this. And this is the thing that we thought would be really important. What it turns out is that the host also does this, okay? And if you go back to a 1968 or 69 Japanese paper, they show this happening. And the tissue what do you think the tissue that they found had the highest activity? Lung, yeah. So where you direct, not liver or even anything else, lung is the best at this. So the lung is activating this drug. So, we were doomed from the outset. So it was lucky we started off with urease, because if we'd have started off with CO, and that took us for ages, and then we'd have done this, I think we'd have just stopped. It would be, this is not a viable thing for us to be spending our time doing. The one we're trying to get funded at the moment is that there are a couple of new TB drugs. There were no new TB drugs for years and years and years. And now people are finally developing new TB drugs. And one of them, or two of them, are these nitroimidazoles, and these are activated by a TB enzyme, and you can measure the 15NO. We haven't done this in breath yet, but this is where we're heading. The problem with this is, how many chemists are in the audience? Okay, so nobody. The intermediate for this, the only way you can get 15 nitrogen in is with a dinitroimidazole. So that's an awful lot of nitro and nitrogen on a very small molecule. And what that usually means is it's known as a high energy compound, which means it explodes. So figuring out how to make an explosive intermediate is particularly challenging. Because we're chemists, we like to see what's going on. We do everything in glass. Okay, so when you have an explosion, unless you've got a decent shield, you're going to get glass in your face, or in your hands or stuff like that. These things usually go off when you move them. It's usually percussion that sets them off. So, yeah, a challenge. But we think it's interesting. And we know resistance is already coming up, and there are a variety of enzymes or systems upstream of this as well that bring about resistance. And so we think that this may actually be a really important, because the last thing we want is to develop these 2 new TB drugs, and then because of crappy utilization, we lose them. We're going to lose every antibiotic. You're going to lose eventually. The bugs will always win. Okay. Thinking that you will get away from resistance is futile. Okay, it will always occur. Star Trek fans should know where that comes from. But can we buy a longer window through screening and looking very, very rapidly? Because if you rely upon growth for this, you're a month in TB, assuming everything goes right. So you've been treating somebody for a month with inappropriate antibiotics at the best possible way. And there are so many potential mutations in this enzyme and the other 4, and it's not an essential enzyme. So it'd be easy from sequence if you see a stop codon, it's in any of those things. It's easy to say, okay, yeah, it'll be resistant. But there are going to be lots of other mutations that you're not going to be able to know from sequence whether it works or not. So this is one of the examples where sequence really doesn't buy you the kind of information you want. So what are these transferable lessons? Well, the specificity of the metabolism is going to drive the specificity of the test, and that should have been really obvious, and it really is. But again, it's something that you learn. So for Yura Gaze, super broad range of microbes, but at least there's no human Yura Gaze, okay? So we're not into those problems of like human pyrazinamidase. It's not as bad as pyrazinamide. And CODH and CAT-G, we can get really, really high specificity, actually, because in the lung, it's only the mycobacteria that we're interested in. I actually gave this talk to people who are really interested in cancer detection, because we know cancer metabolism is different. So they wanted to know, well, can we squirt in metabolites and look for, labeled gases and stuff like that? We'd already tried it, and we haven't been able to get any signal in a rat model of lung cancer that works. But if you're going to look at host metabolism, so if you don't know that this metabolism is strictly microbial, then you're going to have a big increase on the microbes. And for some of the non-isotope breath tests, it may not be clear where it's coming from. Is it the bug? Is it the host? Is it the host only when the bug is there? And there's a host pathogen interaction. So it's a difficulty. The detectability of this metabolism, it's driven by a lot of different factors. They're essentially out of your control. You can only put in as much of this labeled tracer around about isoton as you get isotonicity, and hope that you don't cause irritation or toxicity or stuff like that. And that's really the most you can do. You've turned it up to 11, and there's no more. So you have to have the abundance of the product against other sources, What are the sources? How is your signal diluted? And obviously CO2 is enormously diluted in breath. How fast is the enzyme? How much does it make? Does it make it in all metabolic states? So many different things that you're worried about. Another thing is if your tracer delivery is in a bolus, so a rapid spike directly to the site you're interested in, or is it going to be much slower? So if you use an IV tracer in the lung, it's going to be slower. If you use an oral tracer, absorption is going to be even slower still. So there's a lot on the kinetics of tracer delivery and metabolism that's interesting. Are there other sites of metabolism? And we worry most about the gut microbiome. Most biochemistry is going on in there. Here's some kinetics. Here's somebody with TB. Urea goes in. You see we get a very rapid signal, and it comes down very, very rapidly, okay? And this just shows you bolus delivery is good. I'm aware I'm kind of overrunning. Sampling close to the site of formation is also important. So lung is great. When you go to pylori, obviously the delivery is directly to the site of bacterial infection. But then that CO2 has to be absorbed across the stomach, get around your body as bicarbonate, and then eventually get to your lungs and be excreted as CO2. So that really smears out your signal. So if you're already very, very close to the limit, then that's smearing out over time. Your signal will disappear in the noise. Point of care is good. And for lots of things, you can just use infrared laser detection. The problem with that is you've got to develop a laser for every different analyte. So that can be kind of expensive. So what we really want is more of a mass spec, a portable mass spec. And we're trying to work with some people from the Jet Propulsion Laboratory who make these things to go on spacecraft to try and get it funded. And also editing. I think Jane and I have talked, and lots of people in Jane's group, it's not either or. It isn't all isotopes and just look at one thing or look at the way that Jane works. You can use both. You can use specific metabolism pathways, or you could throw in a sugar and just look at fermentation to look at much broader metabolic pathways. And so you could get an extra dimension in their already three-dimensional data sets. So you could have 4D data. And that might be useful in increasing specificity. Probably not in terms of increasing sensitivity, but that might be really useful. So for instance, a lot of interest in cyanide, exhaled cyanide, and it's potentially a good biomarker for early colonization or early infection with Pseudomonas. But there's lots of sources of cyanide in our body. We actually make a lot of cyanide, or our guts make a lot of cyanide, turn it into thiocyanate. Thiocyanate is present in high concentrations in tissues, and so you've got problems. If we throw in glycine, Pseudomonas just makes it through this way. So can we specifically light up the metabolism from a specific organism or group of organisms? Okay. People here, Seong-won is great. He's done lots of the work. Voyo is an almost Nobel Prize winner. We figured out he was about number 5 or 6 at one time. Worked with a lot on TB. Terry Wu, who's great. He's the Tularemia guy. Runs a very, very strict lab. Zach Sharp and Viorel at the Center for Stable Isotopes, who do all the isotope ratio mass spec. Bill Bishai and Mahmoudou, who did lots of the infection work. NIH for funding, and obviously UNM, Hengame, was absolutely fantastic for this trial. And I'm sorry for slightly running over, slightly too many slides, but thanks for your attention.